Does it look okay? All right. It's really great to be back to Northeastern. And it's been quite a lot of time has passed since we wrote the essay with Dietmar. Many events has happened, have went to different directions. And today, with this lineup of people, I realized that it's really an interesting way to talk about media technologies, the future of semiotics, experience, visualization, meaning of data in so many different ways. I'm really very happy to come to this event and offer my own new insights about where things are heading. So after hearing so many different ways of talking about indexical design, I focus more about not giving you anything about the history or the theory of semiotics, signs, signifiers, and so on. Many have already done this very well. But what I want to talk about is from the perspective of a designer, not a scholar or a historian, but somebody who is rooted in the theory of semiotics in thinking about his own work, to give you an insight about where indexical design is in the realm of the living. So I'm going to look at different embodiments of meaning across different material systems, living and non-living systems, but eventually try to propose a different perspective on the whole conversation of indexical design. So we have talked a lot about indexical design. The word signature only appeared a couple of times, and Simon, he compared fingerprints with signature, and he preferred fingerprints over signature, and we glanced over it. So the word signature is very important because it is the birth of subjectivity. It's the birth of authenticity, and it is a very important element that registers value, meaning, function into the source of signification. So when we look at a document from 1776, everyone is familiar with the Declaration of Independence. It's the signatures make the document speak. So we'll talk about speaking quite a bit, and speaking, what is the realm of the signature versus realm of the material embodiment of the signature. I'll glance over it. But the signatures make the document speak. But then, this is the typical document. There's only 4 of them, apparently, left that are authentically, that can be identified to that historical moment. But last year, somebody actually found a copy in Dallas in the Gilcrease Museum, which is one of the owners of the 4 copies, claim that there's actually a Benjamin Franklin fingerprint on the document itself. Going back to the Jackson Pollock example, can we do spectral analysis? Should we really authenticate it? I don't know where the debate is right now, but there's already a signature of Benjamin Franklin on the document, but now the fingerprint itself is going to make it more valuable because it's double superimposure or super validification, because the living signature and the mark, the semiotic mark, can actually be on the same paper. But my interest is not really to contest the world of fingerprints and so on, because it's been already very well done. I want to go a little bit deeply into the realm of signature, but not into the signature of humans necessarily. I'll touch on that because at some point it's important to talk about it, but the signature of all things. There's a couple of philosophers in the past 200, 300 years who really looked at the signatures that are produced by other organisms, from living things to non-living things. Especially, I'm going to refer to Paracelsus, who wrote a treatise concerning the signature of natural things, all the way dating back to the 16th century, where he really looked at this threshold of when the organism wants to say something, when does the living thing wants to express something, qualities from the inside that are getting manifested from the outside. So I'm going to quote a couple of sentences from Paracelsus, and I'm going to bring Agamben and Foucault into the conversation. But the important thing is to really think about, it's not only humans or an anthropomorphic perspective. Everything wants to say, wants to speak to something else, but the reasons for speaking is very different. So we haven't looked at tigers. We looked at a lot of fingerprints, but we all were human fingerprints. But tigers were also scratching barks of trees for probably thousands of years since their evolution, because they were also trying to communicate with their own kind in terms of showing power, in terms of showing territory, and in terms of claiming ownership of a particular area. But this doesn't mean that tigers were leaving fingerprints for humans to discover knowledge. They had a different kind of causality, because it was only tied to that very particular function. It wasn't about authorizing a document, but it was authorizing place for a very particular time. And especially in this conversation where we think about, should indices be about past or can they be about future, they have a temporal nature, but let's unpack that a little bit more as we go along. But obviously, it's not only tigers. Then you can have spiders to leave marks on their webs themselves. Because if you look at a web, you see these little lines that are actually created to be able to capture prey, but then you see these irrelevant, weird lines that really are not meant to capture anything. The phenomena is explained by natural scientists as stabilimentum, which is about, and the theory says that the signs are actually for other living things, other organisms like birds, or birds especially, or other organisms that can see these spider webs and don't run into them, because it's very costly for the spider to make new ones. Because the spider web is for the prey, not for the bird itself, so birds can avoid by seeing these very, very specific marks that are much more denser and much more figurative in nature. So, going back to Paracelsus. Paracelsus was an astronomer, alchemist, a different kind of scientist of its times who was interested in understanding the inside-outside relationship. He looked at these examples of, how do plants really talk about things? Why do they speak? What is this whole anthropomorphization of the plants from our perspective? Why do we try to see different things in them? And he looks at the understanding of signature from the perspective of the appearance of the plant, trying to say that, okay, if the plant has a particular shape in its appearance, it must be that the plant is actually good for that bodily organ if we consume that plant's juice. So you're looking at euphrasia, a plant which is still commercialized as the label eye bright, which people still consume for, as a medication for their eyes for better clarity, and apparently it looks like an eye. Of course, if you heavily weigh on the scientific methodology on it, you will realize that that causal relationship really has no meaning in a particular perspective, but we still, from even in Paracelsus times, he still finds some meaning in that association, because he thinks that signatures are not necessarily just creating facts, but they're actually establishing the context of interpreting these facts. So they are basically the code that really justifies the relationship between signs and signifiers. So another irrelevant example, you see we consume walnut, you think that it's going to be for your brain, but apparently, some people still believe in it. But again, the appearance is still constructed by the appearance of the walnut is already constructed by the gaze of the humans that are consuming it, and then eventually relating it to the brain. Doesn't make that much sense today. But the signature, the realm of the signature is, in the 16th century, was very different, right? And we are talking about the authentication of paintings, the authentication of resemblance, who creates which painting, how does he authorize something. The notion of authority and the notion of the hand in the image, the notion of me signing my own word, my own image, has a lot of history in human communication. There's no doubt about that. But Paracelsus was actually smart about acknowledging that as well. So there are different kinds of signatures in the world, in the living world, across different organisms, but they're establishing the type of epistemologies that we should be utilizing in evaluating them. So the evaluation of the plant sign is very different than the signature of Titian at this point, because they operate in different epistemological contexts. So for him, signature is an index in itself. So we are not talking about the physical indexicality here. We're talking about the signature that registers the sign, the function of the sign. It creates a function of existence for the sign, so the sign can actually exist in language, exist in reality, exist in material form, so its embodiment can actually vary across many different contexts. And the important part that what we do today, the study of signs, signatura, is actually the act of deciphering the signs at the same time. So through deciphering the meaning of these signs, we actually create them in a certain point, and we actually create knowledge out of the interpretation process. So we make the knowledge both by making the signs and interpreting the signs through the signature, because it registers the domain and to which how they can be interpreted or how they should be interpreted. So what I'll try to do, to speed up obviously a little bit, but to talk about a number of different domains, from very small to a little bit towards more bigger things, to be able to really come up with a variety of different signatures that we design, not only see and observe from nature or interpret from nature, but actually we design as material embodiments, so that we can bring back again the notion of value, meaning, authority, ideology, significance, experience, back into the conversation of the physical. We are not talking about the language or the speech that the histories and theories of hard and soft humanities have been built on, but going back to the physical notion of sign making is a measure of existence. And I will talk about what do I mean by performativity and response to performative theory and so on at the end, but I want you to keep thinking about what a signature is, a way of making signs that actually exist in the real world. And the measure of existence is an important thing. So I have a little video, maybe I can, hopefully it will play. Let's see. So let's see. So you're looking at a screen, my little screen, which is showing a little droplet, purely chemical substance that can solve a maze. So you drop a little liquid here and another liquid there, and then it goes and finds the maze. There's no organism, there's nothing in there that is, scripted, no computers, no machines. It's just a chemical process. Something goes from one place to another using its chemical agency. Because there's a chemical signature in the environment where there's a pH difference between one point and another point, so that the molecule figures out the path by itself to be able to go from one place to another. And this phenomena is important because we have been studying signs or signatures of life for quite some time, and I'm especially interested in that conversation. What do we call living? What do we call not living? Requires a lot of thinking in terms of signatures of what we think living things leave behind, and what kind of things that actually needs to go into a living, call them living. And this is not a droplet, this is now a little cell. We call them protocells. They're little bags of molecules coming together which function like living. So this molecule can leave the traces of living artifacts. For example, you can incorporate in it DNA so that it can make proteins. So you can leave certain traces of proteins that, as if a living organism is producing them. So you can create these things on the bench and actually make them produce chemicals that can be visible or detectable or measurable as if they are created by living organisms themselves. So, and then what you do with them, some people use them in cancer research, some people use them for asking more questions about the origins of life, but the important thing is they leave a signature in our epistemological constructions right now, because they pose a limit on what they pose a limit on our ability create life from scratch, from known parts. Scaling up a little bit more, you're looking at now cells, tissues, made of cells that are coming from real organisms. So what does it mean for working with living organisms so that they come together and form traces of life or traces of vitality? Because what living things do, they have signatures of themselves. They know they know that each other's difference, so they can detect each other in an environment. They can come together and start differentiating. So there's signaling mechanisms between tissue cells which allow them to come together and differentiate so that they become something totally different. So this very complex phenomena that is happening in our muscles, these are heart muscle cells coming from a mouse cell line, is actually an interesting thing for artists to explore, because you can utilize the muscle tissues themselves to construct lifelike structures, like lifelike creations that actually problematize the notion of where the boundary between a mouse cell or a pig cell or a human cell blur, so that you can really create these chimera, weird, problematic artifacts that can look like wings, but they're actually created from pig cells. So this is an artwork that I'm referring to, Orhan Katz and Yanat Zur, quite an old project, but a good statement in terms of how you really construct with some other organism another one. So the pig cells become a signature for pig itself, but they can totally stand for something else, so in the hands of a human creator. Going one more step up, now I'm going to talk a little bit about organisms themselves, because we have been getting very much better in manipulating bacteria, yeast, and other kind of microorganisms. And this is an old artwork. On the right, it looks like a 4-year-old kid drawing, but it's actually Alexander Fleming himself making the first germ paintings on a bench. He not only invented, he had an amateur art training, so he was really very much into painting, but probably his parents didn't allow him to go to the art school, like nobody does. So he ended up creating the ability to control life and death of bacteria so that he can make them live at a certain amount of time so that they can appear in certain areas, make different figures, and so on. The artworks, depending on your taste, may actually become very, very attractive or terrible looking, but his ability to control the duration of a lifespan of a bacteria allowed him to paint by them. So this is an important phenomenon. But of course, we moved on many, many years after this. So one is working with designer bacteria, designer organisms, and we don't really work with them on dishes, petri dishes like this. We just work with their representations. So we consult back to the whole symbolic world to be able to design these indexical organisms. Indexical in the sense that they're physical, they have a color, they have a materiality, they have a smell, and they assert themselves into the real world, and eventually become artifacts that are patentable, that become cell lines by themselves. This is a yeast that is already, it's in public domain, but a different version of it is patented for bringing beta carotene, vitamin A, into bread and other food kinds, so that you can, if you serve them for kids that have vitamin A deficiency, that can have actually a lot of nutritious value. So it is a big public project that is, that is actually supported by the White House, so that you can make these loaves of bread and distribute them in Africa or in places that are in need of vitamin deficiency. So going up in the ladder of organisms, we talked a lot about fingerprints, but obviously most of you will know about the Human Genome Project. Once we advanced our ability to decode the human DNA, it became an important way of identifying humans with a certain trace. So DNA became the center of the discussion about whether we are results of our blueprints, or are we really, are we really shaped by the environment. Today we already know these answers, or most of it, but it's a history that started from 1950s onwards. The schematic shows from 1990s, but it's a very, very rich history. So one more time you see O.J. Simpson's DNA, but I'm really not going to talk about this, because the importance is the creation, the design of the genes themselves are a bit different from reading somebody else's existing genetic markup. Because the plasticity of the manipulation, as Paulus Volonus's work has demonstrated here many, many times, is about the image of the DNA. But the DNA itself is also a plastic entity that is manipulated on many, many times, before even looking at the image itself, it can still function as a trace or a denominator of a certain characteristic. So the DNA, why is DNA so important? Because now with the advances in genetic engineering, genetic sequencing, we can identify the microbial signature of places. So instead of, this which is the equivalent of fingerprinting places based on the bacteria that are living in those places, by collecting samples from certain places, you can identify what kind of people live in those places, because people leave these bacteria around there, and eventually characterize the function of these places. It's where, what is the nature of these places, because bacteria use humans to go from place to places. But this is obviously not a very, this is also a very political case. We'll hear about microbial signatures quite a bit, so I'm not going to dwell on that too much, but I will just an anecdote which happened on Penn's campus apparently last year, when there was a big metagenomics project where, close to 100 students went all around the campus and started sampling the places, and at some point it created a big social political problem, because students of color complained to the department saying that actually some white female or male students were going after the places that they were living and swabbing the places to find out what kind of bacteria they were leaving behind. So students obviously were not trying to do that, but apparently the word germ, it was a big problem, because no one uses the word bacteria or microbe. People use bacteria or microbe in research papers, but when a publication comes out and says that, well, students mapped the germs on campus, the students of color realize that, well, maybe they're after, they think that we are actually much more dirtier than the others. It was a public outcry, and it was very difficult for the school to really handle it. But apparently, it was solved because it was a communication problem, because the students really didn't intend it, things like that. But by searching a certain place, by trying to find a particular signature in that place, you already make a political statement. I also want to touch a little bit on algorithms, because it is important to bring back the machine into the picture, because it's important to look back and see where the semiotic is really actually functioning today most effectively, most ideologically, most politically, because all of the algorithms that are actually having some impact on our life are based on our data, or the traces of our data, or the signatures of the data that we leave behind. Here I'm not using the word signature metaphorically, because we leave literally by our search histories, by what we listen, where we go. We actually train these algorithms, so they look after our signatures. Literally go after what kind of things that we leave behind, intentionally and not intentionally. So you're looking at algorithms that are trained to decipher signatures. So when you write your zip code on an envelope and send it away, you know that the post office will decipher it, because there's smart algorithms that figure them out. But actually, they can look into your signature and really figure out whether you signed it or not. In certain cases, it's useful for people to figure out forgery. And going back to painting itself, we love paintings. This is a Rembrandt portrait, self-portrait, that was painted in 2016. So researchers from University of Delft last year announced this beautiful Rembrandt portrait, which was 3D printed, 3D painted. That's a thing. It's a beautiful video. I encourage you to look at it. It's based on 168,000 fragments collected from different Rembrandt paintings, that were inputted into a machine algorithm, which clearly figured out how Rembrandt's impressions, brushstroke impressions, were made. So he calculated based on the other kind of factors that he was using in terms of making his own depiction, because it's a self-portrait, and making a self-portrait of portrait is really a difficult thing, but it's a bold act, and it looks pretty much like Rembrandt, if you ask me. Going back to the conversation of speaking. So Agamben reads Foucault when he's looking at the signature of all things, Paracelsus. Foucault says something very smart. Foucault says that speaking is really not about, communicating one's ideas. It's about doing something. There's a certain kind of action and intention and ideology involved with it. It may come across pretty normal, but I want you to think a little bit more about meaning. When we think about meaning, we think about certain kind of interpretation, there's a certain kind of, there's a desire to be understood. But before the desire to be understood comes into place, there's a desire for action. So when you speak, you transform something. So what does this mean? So going back to cells. So when you really have cells to speak about gender, you can today go to any in vitro fertilization clinic and actually pick the genders of your kids. You can actually, artificially inseminate cells, and you can pick gender, and if you really, really want to go through it, you can sequence the genes, and you can really pick what you want to have, what kind of offspring you want to have in the future. So the cells speak certain characteristics of your offspring, but by choosing by making that selection, by choosing which signature you like, you actually have a control over the process. So we not only design our own kits, but we also design mosquitoes themselves. So you're looking at, well, we talked about flies, so I had to show a mosquito in the presentation. Oxitec, OX513A, which has FDA approval in Keys of Florida about the yellow fever epidemic. Brazil has already approved it also for Zika virus in Brazil. And actually, I think also in a couple of other places in Latin America, maybe even France itself. So this is a mosquito, a designer mosquito that is designed to die or designed to, make its future generations be more fragile so that they can kill in a couple of generations. So it mates with a female, and the female becomes weaker, and eventually, the generation unfolds. So it's better than, some people think that it's better than using mosquito killing, mosquito repellents, or, or just killing the mosquito by chemical means. It's building a suicide mechanism into the organism itself, so it dies by itself. So this is the place where we really think about, okay, the mosquitoes detect each other's signatures, and then you use this signature to be able to kill the organism. Going back to a little bit of my own work, I'm interested in this whole idea of creating protocells. I'm interested in the idea of creating living organisms, but I'm also interested in pushing the idea of signature, not my signature, but pushing this idea of machines creating their own signatures, machines creating their technologies, because there's a which is not so different from you picking your own kid when the system proposes you a bunch of different solutions from gender selection to other things, and then you make your decisions. But if I use a machine, if I build a machine to be able to generate variations of a different organism, how will it actually evolve to different, what will that organism look like, what will it do, what kind of intentions and ideologies it will have, because signatures are tied to intention and ideology. It's very different for when they're created by a machine which doesn't have the same kind of evolutionary reasons to survive or meet certain criteria that we call, survival of the fittest and so on. So you can really unpack this whole narrative of evolution, design, and so on by unpacking the notion of signature or creating things that can sign themselves as organisms. So this is a little machine that genetically modifies organisms so that you can create different kinds of, you can use different kinds of design methods, combinatorial design methods and parametric design methods to, it's a little bit skewed, to design bacteria. I will later on, so you load up DNA and bacteria on one end, and you take the organisms on the other hand. I showed a basic version of that machine in the exhibition that Jans curated a couple of months ago, Wetware, which was in UC Irvine. So basically, you introduce DNA and bacteria into an environment in standardized capsules. You apply electricity to them, and then eventually the bacteria gets transformed into the organism. You grow the organisms, different variations of the organisms at the same place, and eventually allow the machine to do new kinds of variations that you may not think about. So why is this important? I wanted the machine to be able to explore the idea of what a banana may look like in the future, because bananas are going extinct Cavendish banana, which is the popularly consumed banana today, are threatened by a fungal disease. They are very well produced all around the world right now, but they may eventually go out of, they may go extinct because there's no really known solution for the fungus. And what people are doing today is when the fungus hits the plantation, they close the plantation and open a new one. There's no fungicide that will, that will kill the fungus itself. So what is the problem here? The banana itself has a signature for being itself, but eventually that signature is going to die. The Cavendish banana cannot really survive on its own because we've been cloning the banana for the past 50 years since we have started planting it. So the things that can sign themselves have also a capacity to change their signatures to something else, but the Cavendish banana cannot do this because it's been a clone. So when you clone it, you always repeat its own signature. So the machine itself has produced different variations of components that make up the banana taste. For example, it has DNA that can make a variation of the sweetness of that banana. It has a variation on that flavor for that banana. So it smells like banana, it passes like banana, but it will not look like banana. So what does it mean for this? You can actually use bacteria to produce the elements of the banana which actually cannot be replicated by the banana itself. But what does this mean? So you can have a lot of different things. Will it look like the picture of a banana? Because it is, in appearance, it doesn't look like a banana, but it is also, is it signed by these organisms? It has a certain kind of authenticity because they are one of a kind. They have a certain kind of, they are not coming from nature, but they're actually, things that are created by other organisms. So they have a certain kind of subjectivity. The one on the left is probably less authentic because it's an industrially produced cloned banana. It cannot vary itself. These are the bananas that the bacteria has grown in different form factors. The little capsules that you see there in here that have the bacteria that actually make up the different components of the taste. So if you take these things and put inside a blender and blend it up and then put inside your milk, you will not feel the difference between a real banana and a fake banana. So in closing, I want to talk a little bit about going back to I want to revisit the whole idea of the signature, because I have shown you a bunch of different embodiments for signature, from protocells to the pig wings, and the signature that is marked itself, because there's a difference between claiming that something is a signature, this is a signature that is signed by the organism itself, or the human itself, or the machine itself, but that is okay. But Foucault gives us one more element about thinking about signatures as statements themselves. So signatures are not just something that authorizes a statement. If you think of a painting as a statement, the signature is not the element that authorizes the painting. The signature itself is the statement itself. Because by asserting itself into the realm of the painting, or the language, or chemistry, or the biology of the organism, the signature, whether it's the DNA, or it's the cell, or it's the organism, it asserts, it gives a capacity of aliveness. And that capacity is an unknown capacity, because it, unlike machines, or unlike, performance that is prescribed by a certain known agenda, we don't really know what aliveness really means for living things, because they don't necessarily always evolve and go into different directions. So it's this unknown element that makes the living signature quite different from a signature made by humans or made by machines. So as designers, our role maybe is giving different embodiments of these matters, but also allowing these embodiments to actually speak about their own existences, so they have their own agencies, they can go on their own, to go on their own unknown, unpredictable futures. But also thinking that existence is really not only performance, it's not only agency, it's not really about just meaning, because it's also about this unknown, this ability to not being decided, not not being known, not being reduced to an epistemic category, because they really actually, out there existing for themselves. So we have been really looking at maybe signatures in a very mechanized perspective, because we have a very anthropomorphic point of view, but it's really important to step back a little bit and really look at the ability to leave the signature alive. Maybe it is going to become something else in the future, or maybe it will go back and resemble something else that has been coming from the past, but letting the signature to be itself as a in itself, so that it doesn't become a signifier or a signified or a context or an icon or a symbol, but a thing that it's imminent to its own performance or existence. So this is how I will finish my little introduction to this session. We will have many nice other speakers who will talk about different aspects of what I have talked about and maybe not talked about, but thank you for your attention.